Chapter 13, Starting Man in a Test Tube. All hell will break loose, politically and morally, all over the world. James Watson, biologist, in forecasting the reaction to the placement of laboratory-grown embryos in women's wombs. The development Watson warned us against is best known to scientists as in vitro fertilization followed by embryo transplant. What this means is that the human egg is fertilized by a sperm in vitro, or in glass, in a laboratory. Then after the tiny embryo has grown for several days, it is implanted in the womb of a female. There it continues growing in natural gestation. As Watson predicted, hell has indeed broken loose. As distinguished obstetrician lost his job at the Columbia Presbyterian Medical Center in New York City for going too far in helping a woman have a baby fertilized in his laboratory. A $1.5 million lawsuit was brought against an official of the hospital by the woman and her husband, an Italian scientist who claimed to have fertilized a human embryo and to have let it keep growing in his laboratory for some weeks was asked by religious authorities to discontinue his work. He did so, at least inside Italy. Some wanted to prosecute him for murder for terminating the life of an embryo. A world-famous British physiologist has been severely criticized by some of his colleagues for his alleged insensitivity to the implications of what he has been doing. A number of experimenters to escape the pressures to stop experimenting have reportedly been going underground in order to continue their research. In early July 1974, while I was attending a conference on biomedical ethics at Manhattan College in Purchase, New York, I heard the startling news from a noted Chicago anesthesiologist that Russian scientists had brought to natural birth 3 infants who had begun life as a result of laboratory manipulation of human seed. This was the first actual birth I had heard of that had started in a laboratory dish. He added that according to a report he had heard previously, the Russians at one point had 218 fetuses growing which had been fertilized in vitro. 2 weeks later, I read that England was in something of an uproar because of seemingly casual announcement by a noted doctor. He made it in press release form before presenting a formal paper to the British Medical Association on the general aspects of embryo research. The doctor was Douglas Beavis, a professor of obstetrics and gynecology at Leeds University Hospital. Until then, he was best known as an expert on biochemistry of the female reproductive organs during the first weeks of pregnancy. Without implication, the gaunt, mild-mannered Beavis announced that 3 babies conceived in a laboratory had been born in a natural way. The infants were then between a year and 18 months old. They appeared to be normal. Under probing it, he indicated that at least one was in England. He also intimated that he had a hand in managing their conception and the transfer of the embryos. Although he reported success with these 3, he said that 30 others had been failures. What happened to them? Were any born as monsters or aborted as monsters? Or didn't they survive the transfer to the womb? The answers to these questions were not forthcoming. Within a week of his announcement, the Medical Research Council in Great Britain called for a halt to research with human embryos until the serious ethical problems involved could be studied. Meanwhile, the editors of the Journal of American Medical Association had asked for a moratorium on experimentation with in vitro babies-to-be. A British Sunday newspaper reportedly offered Beavis approximately $70,000 for an exclusive story on the test tube babies. Beavis complained that all the publicity had made it possible for him to continue his work with embryo transfer. Many of his British colleagues berated him for not making a proper scientific report to permit evaluation of his claim. Some expressed skepticism, others outrage. Beavis said he felt impelled to withhold details in order to protect the privacy of the three mothers, but added that in due course he would publish an official report of the experiments. The announcement by Beavis came as quite a shock to scientists in several countries who had been working on test tube fertilization and embryo transfer. Beavis had not been counted among the frontrunners, although it was known that he had been working in the field. Two of the countrymen, R.G. Edwards, a physiologist at Cambridge University, and his working partner, Patrick Steptoe, an obstetrician at Oldham General Hospital, were far better known. They had had massive funding. They had had worked with hundreds of women volunteers, and their efforts had received wide attention. Steptoe made a critical and skeptical statement when Beavis announced his feat. Edwards, Steptoe, and their associates were in a position to know the magnitude of the problem. Achieving in vitro fertilization and then successfully transferring the embryo to the womb is an extremely formidable enterprise. Here are some of the problems. The first is to obtain a human egg that will be used. The egg must be just ready to leave the ovary and start the slow trip down a fallopian tube. In early human experiments, the egg was taken from a fresh cadaver or during a gynecological operation in which the ovary was opened. A two-step process developed by Steptoe and Edwards in the 1960s solved the problem more simply. Sex hormones were introduced into the women. As a result, a bumper crop of a dozen or more near-ripe eggs were produced in the ovary. Then Steptoe went in after the eggs by making two tiny slits through the woman's abdomen to an ovary. Into one slit went a long, slim, illuminating telescope. Into the other went a slender suction device. Several eggs could be siphoned out and put in tiny laboratory dishes. Even though you surround the egg with thousands of sperms, it will not be fertilized until the sperm destined to succeed has been exposed to a chemical found in the fallopian tube of the female's reproductive tract. This was a finding of M.C. Chang's at the Worcester Foundation for Experimental Biology in Massachusetts. When fertilization occurs, the cells will cleave and multiply only if the nutrients are precisely right. They must be precisely right hour by hour as the biochemistry of the developing embryo changes. Before the new growing embryo can be transferred to the woman's womb with any chance of success, it must be checked for chromosome normality. This requires manipulation that can cause damage. The transfer itself, which also obviously involves manipulation, must be geared precisely to the time the fertilized egg would reach the womb if it had been able to grow down the fallopian tube. Normally, the egg takes 5 days to make the trip. The embryo must attach itself securely to the life-giving lining of the womb before the flushing out of the menstruation occurs. This has proved to be a major hurdle. The question arises, why take on all these challenges and hazards when nature's way has worked fairly well for millions of years? Some investigators explain that they are simply searching for more knowledge about the conception, that in the process they hope to obtain clues to simpler, less troublesome contraception. An ideal contraception might be an injection of antibodies that would surround the ovulated egg and prevent penetration by the sperm. In July 1976, proceedings of the National Academy of Sciences indicated that animal studies support the feasibility of this approach. The research in test tube fertilization and embryo transfer is more advanced with animals than with humans. Pioneers in the laboratory creation of potential humans. The British team of Edwards, Steptoe, and their associates is generally given the credit for achieving in vitro fertilization in 1969. They are given the credit primarily because of their massive documentation of the early stages of fertilization, but a number of possible or probable successes occurred before then. As early as 1944, John Rock, the American co-developer of the birth control pill, attempted test tube fertilization as a part of his research on sperm and egg behavior. In a few cases, he reported observing a sperm apparently fertilizing an egg, and later seeing the egg actually dividing. M.C. Chang raised a question about this. He pointed out that if you take a human egg out of the body and leave it alone in a culture dish, it will, as part of the process of dying, show signs of dividing. In 1959, a confusing series of events began in Italy and continued in Russia. A physiologist at the University of Bologna, Daniele Petrucci, reported that after 40 failures, he had fertilized a human egg in vitro and had kept the embryo alive for several weeks artificially. He apparently did not try to implant his creation in a mother's womb. Many scientists were skeptical of Petrucci's claim to success, but he was taken seriously enough to be called into conference at the Vatican and requested as a good Catholic to stop his experiments. He did announce a halt a few months later, but still later he was invited twice to Moscow to confer at length with officials of the Institute of Experimental Biology. They too were working on the test tube fertilization of babies. Most solid claim to have beaten the British team of Edwards, Steptoe, and their colleagues could be posted by the obstetrician Landrum B. Shuttles in New York. Shuttles is one of the giant figures of reproductive biology. Physically, he is no giant, but a wispy, amiable man of modest origins and blunt speech. In conversation, he calls gonorrhea by its earthy, more familiar name, and he speaks of Edwards with something less than reverence. Shuttles quite certainly beat the British team in the follow-up of transferring the lining of a mother's womb, a human embryo that had been fertilized in a test tube. But what about the actual fertilization? For 27 years, Shuttles was a professor in the College of Physicians and Surgeons of Columbia University. He told me that by 1951, he had achieved in vitro fertilization. During the 1950s, he took phased microphotographs of laboratory-created embryos that convinced many scientists that he had succeeded in growing a multi-celled human embryo in his laboratory. His pictures had been included in some 75 medical textbooks and were displayed, greatly enlarged, at the American Museum of Natural History. The pictures have remained on display there and can be seen at a number of other museums, including the Academy of Science in Moscow. They certainly look impressive to my naive eyes. But some specialists were not convinced. There was the possibility that the egg could have been simulated into cleaving and multiplying by some chance factor without the aid of a male sperm. This is called parthenogenesis, or virgin birth. It is not miraculous in nature. According to one report, as early as 1963, Shuttles was experimentally implanted test tube grown embryos in women. He mentioned to me that around 1968, he felt he had fully mastered implanting. He said he took a picture of the implant in the woman's womb, but kept it quiet. Implanting was already a touchy subject. In January 1971, the British journal Nature, one of the world's most respected scientific journals, carried a detailed report with photographs by Shuttles on his experience in collecting and fertilizing a human egg in vitro and growing in culture until it is ready for implant. It is ready after 5 days, when it has become a 100-cell cluster. An embryo at this critical point is ball-shaped and known as a blastocyst. He said that the reason implantation did not follow was that women involved from whom the egg had been taken was recovering from an extensive gynecological operation. He may have deliberately set up the project so that it would stop short of implementation. There was controversy among his own colleagues on the wisdom of attempting it. Later that year, an American publication reported that Shuttles had repeated the feat and then had plunged on and implanted an embryo in a woman. The implant was reportedly placed in a woman, not the natural mother, who was scheduled for a hysterectomy 2 days later. The operation was duly performed and the embryo appeared to be growing normally. Later, Shuttles described the identical implanting during a symposium. An account of this symposium was published in November 1973 issue of the Journal of Reproductive Medicine. He said that the embryo was nidating properly. It consisted of several hundred cells at this point, and no contraindications for continued development were discernible. The menstrual cycles of the two women involved had been synchronized with hormones. 2 days, of course, do not constitute a full-blown pregnancy. All the documentation I have assembled indicates that regardless of who gets credit for first in vitro fertilization, Shuttles had been at least one first. He apparently was the first researcher in the Western world to report the implant in a womb of an embryo created by in vitro fertilization. What Russians were doing is perhaps another matter. In 1973, Shuttle's pioneering got him into a mess of trouble. While still at the Columbian Presbyterian Medical Center, he sought to help an infertile woman from Florida have a baby. Her fallopian tubes were blocked. He arranged with the surgeon to obtain one of her eggs and expose it in vitro to her husband's sperm. Soon thereafter, according to Shuttles, his superior at the hospital confiscated the specimen. Implantation became out of the question. The superior's explanation was that while it was all right to experiment with fertilization, it was much too early to try implantation of an embryo that could grow to term and be born naturally. There had been too little work on primates to establish what the risk of malformation was. And further, he added, how do you get informed consent from a fetus? The Code of Medical Experimenters requires, though the wording is imprecise, that permission must be given by a human subject before experimentation can be done. Is a fetus a human? If it is, at what point did it become so? Obviously, with the fetus, consent can come only from the parents. Shuttles felt impelled to resign. Subsequently, the couple involved brought suit against the hospital on the grounds that without their consent, he had deprived them of the chance to have a baby. At this writing, the case is scheduled for trial soon, with Shuttles as a key witness. Shuttles then became director of research at the New York Fertility Research Foundation. Since July 1975, he has been continuing his experimentation at the Gifford Memorial Hospital in Randolph, Vermont, where he is chief of obstetrics and gynecology. I heard a doctor publicly state at a conference that Shuttles had gone underground in embryo research. When I raised the question of Shuttles' activities in embryo research while he was still at the Fertility Research Foundation, the doctor murmured, word of his presence at the Vermont hospital has spread, and the hospital has become a minor mecca for infertile women. He has been assured that he can proceed with his work on in vitro fertilization and embryo transplant without constraint. Shortly before this book went to press, Shuttles told me that he was just about ready to try to help an infertile young wife have a baby by a revolutionary new method. What did he think of the Beavis announcement in 1974 about those three apparently normal test tube babies? He said that from what he had seen from his own work with more than 2,000 human eggs, he had no doubt that Beavis and possibly others in the world had now done it. Then he mused, if I had gone ahead back in the 1950s and 60s, I would have had the cat skinned before the current uproar. Meanwhile, in 1973, an 8-person team at the Queen Victoria Hospital in Melbourne, Australia, implanted a very young embryo in a woman, and it apparently remained viable for 9 days. By that same year, the Edwards Group in England had succeeded in getting eggs fertilized in vitro and to grow in wombs, but for no longer than 21 days. 4 months before Beavis startled the scientific world by announcing the 3 natural human births, the Quarterly Review of Biology carried this discouraged statement from the usually bold and coolly confident Edwards. At present, there seems no immediate chance of any serious attempt to grow human embryos well beyond the blastocyst stage through implantation in vitro, and any claims to have done so can clearly be dismissed. Of course, it is possible that the babies claimed by Beavis will not withstand scientific scrutiny if it becomes possible. One cautious speculation is that he may himself have been deceived. Women are sterile for reasons other than blocked tubes. One suggestion is that if he obtained his eggs by using hormones to grow a crop of near ripe eggs in the ovaries, hormones may somehow have overcome the 3 women's sterile condition. In this case, just possibly, and despite the implants, their babies could be a result of normal pregnancies following intercourse. That explanation seems to require a lot of coincidence, but only time will tell. Legal aspects of starting human life in a test tube. The Vatican has taken the position that human life starts with the fertilization of the one-cell human egg. Anything a scientist did to terminate a test tube experiment on the viable embryo thus would be illicit and theoretically leave the scientist open to charges of homicide. It is doubtful that any civil prosecutor would ever bring a scientist to court for terminating the 16-cell speck he had grown in his laboratory, even though the speck contained the blueprint for a human life. After 3 months of gestation, when the embryo becomes known as fetus and he has taken on visible human form, it might well be entitled to some legal protection. There are laws in some states against fetal experimentation. If a defective human, not to mention a monster, is born as a result of an experiment, the scientist might also be vulnerable to the damage suit from the parents, and possibly the institution funding the experiment might be vulnerable. Edwards, who has understandably looked into matter carefully, seems to feel moderately secure under British law. The parents bringing the suit would have to prove that something the doctor-scientist did was the direct cause of the defect. Expert witnesses presumably would come forward to testify that any risk assumed was not unreasonable. Edwards mentioned that in some countries, including the United States, the burden of proof shifts to the defendants if the case involves ultra-hazardous activity. He also mentioned as a conceivable hazard that a plaintiff might charge that the experiment was performed without sufficient prior experiments on animals. But he discounted this by saying that the work already carried out on animals would appear to make this argument irrelevant. Since virtually no work has been done on non-human primates, that seems as astonishing position. If the customary scientific approach had been followed, at least several dozen monkeys and 20 chimpanzees would have been born as a result of test tube fertilization before experiments with human embryos were undertaken. And once born, the monkeys and chimpanzees and their offspring would have been studied rigorously for abnormalities, including mental retardation. A French commentator on the legal aspects of human embryo transfer offered this equally astonishing opinion. At present, the risk to the fetus is neither known nor capable of evaluation. Therefore, we believe that the doctor is not professionally responsible if a child is born malformed. This seems to hold that the doctor is innocent because he is ignorant. In the United States, a proposed guideline for institutions seeking federal funds from the National Institutes of Health had this to say about products of in vitro fertilization. No research involving implantation of human ova which have been fertilized in vitro shall be approved until the safety of the technique has been demonstrated as far as possible in subhuman primates, and the responsibilities of the donor and recipient parents and of research institutions and personnel have been established. That is not law. But since many United States institutions involved in early life research seek financial help from NIH, it has stimulated some prudence. The various national commissions should seek as far as possible to arrive at an international consensus on whether to proceed, and if so, how. The geneticist James Watson, who predicted that all hell would break loose, has expressed grave concern about uncontrolled embryo research. He estimates that it is technically foreseeable that some kinds of in vitro manipulation of human eggs could become medical practice in many major countries within a decade or so. He proposes international agreements to limit such research before the cat is totally out of the bag. Presumably, he was referring to the same cat that Shuttles was going to skin. Such steps as national commissions and international agreements, it seems to me, are the least that humanity should expect of us. Chapter 14, Leased Wombs, Artificial Wombs, Non-Human Wombs. If children can be had without being born, working mothers need not to be affected by childbirth. This is happy news for women. The Chinese communists were reacting to the news out of Italy that the test tube growth of human embryos had proceeded to the heartbeat stage. The Russians were equally intrigued. Both societies had placed great emphasis on the idea that everybody, men and women alike, should engage in productive labor. To such societies and technology that offer to reduce the woman years lost because of pregnancy is interesting news indeed. In Russia, for example, more than 80% of all women of working age have jobs. Aldous Huxley's fantasy of growing babies in central hatcheries is technically almost halfway feasible already. Meanwhile, another technique emerging from embryo research could soon make possible the use of substitute baby bearers. The technique might prove to be more appealing to liberated Western women. The communists may be less interested in it because it does not relieve the problem of lost woman years. Still, there may be high cost factors of mechanical incubators to consider. If the techniques are permitted to proceed, it would be interesting to speculate which system, by the year 2000, would offer the cheaper product, that is, baby, and which system would offer the higher quality product, again, baby. Using apes or cows to gestate humans to be might turn out to be the cheapest of all. The possibility of transplanting an embryo from one human to another does not strike quite as many sparks as the test tube fertilizing of human sperm and eggs. It doesn't have the awesome potential for moving on to novel forms of reproduction in vitro creation of potential human has. Transplant to non-humans would be pretty controversial, however. The shuttle's concept of using another woman's reproductive apparatus for a week or two in order to bypass a cause of fertility in the wife, such as a clogged fallopian tube, might generally be viewed as preferable to adoption. The use of mercenary mothers to take over childbearing for working wives might well have substantial appeal, especially if current trend towards careers for married women continues far into the future. At first glance, the use of surrogates seems like a logical evolution. On closer inspection, though, it seems controversial at best. This is so whether the hired mother acquires the embryo by transfer from the wife or by artificial insemination, in which the husband's semen is used. Mercenary mothering would threaten to weaken both marriage and the family. The wedding vow is based on an assumption of exclusivity in all matters relating to planting seed and bearing children. We would have to change our concepts of what a family is and broaden the definitions of such words as father, mother, parent, motherhood. How important is childbearing to the self-fulfillment of women? Have we overrated it because they have had to do it anyway? Or is it an important part of women's concept of a complete life? Can married women develop the love and sacrifice and dedication required to rear a child successfully if they deliberately abstain from bearing it? Would there be a diffusion of identity and uncertainty about what it means to be a son or daughter or a parent anymore? Would mass educational programs be introduced to make the young less concerned about bloodline identity? We don't know really. A few years ago, the British Medical Association proposed that surrogate motherhood not be attempted until much more study had been given to probable psychological demands on the wife, the child, and the hired mother. Still, another question is whether thinking of the fetus as a piece of property to be paid for in a cash on delivery would contribute to the dehumanizing of man. And with the mercenary mother, it is not probable that authoritarian regimes would start singling out physically and perhaps mentally superior girls and forcing them into careers as baby bearers. Certainly, such questions make it all the more imperative that national commissions to regulate reproductive research has been established. Research on the ways to sustain life outside the womb has taken an interesting turn to the study of marsupials. The animals which lack a placenta include the opossum, the kangaroo, and the wombat. At a very early stage in gestation, the tiny immature marsupial is discharged from the mother's womb. Somehow it manages to climb into the mother's snug pouch where it clamps its mouth onto a substitute for a placenta. An opossum is the size of a cat, yet a dozen of a newborn could fit into a ping pong ball. Scientists in Van Nuys, California, have been deceiving newborn possums into attaching themselves to nipples inside a plastic transparent pouch. Various nutrients and drugs are fed through the nipples, and then reactions of the baby opossums are studied. In the Huxley's Imaginary Laboratories, the baby makers varied nutrients depending on what brand of humans the regime wanted to produce at that particular time. The French biologist Jean Rostand saw the kangaroo's reproductive pattern as a possible model for humans, since in humans the early weeks of gestation are proving to be by far the most difficult to manage outside the womb. They are the most error prone. He suggested that we adopt the marsupial approach. Human mother would undergo a normal pregnancy for a few months. At that point, she could be chemically stimulated to go into labor and discharge the fetus. Fetus would go into an artificial womb or incubator. An alternative would be to surgically lift out the fetus. At present, scientists are working from both ends of the human gestation period to reduce the time the fetus needs to be in the womb. They assume that by a point in the future, the original 9-month period will have shrunk to 0 days. They are making spectacular progress in being able to save prematurely born infants at earlier ages. Vincent Collins, chief of anesthesiology at Cook County Hospital in Illinois, told me, "We can now take over a fetus at 5 months, and soon I believe we can do it at 4.5 months. We can keep 1 pound baby alive outside of the mother." 4.5 months is half the gestation period. However, at the other end, the early embryonic phase of gestation, progress is measured in hours, at least in the Western world, and the hazards of defects and deformity in the first days and weeks are vastly greater. All this suggestion, all this suggests to me that the drive to create a completely artificial womb is folly and should be brought under the social control. It is folly just as the dream of colonizing the moon is folly. It could even eventually be done, but at considerable moral, social, psychological, and economic cost. Thousands of near-term fetuses and perhaps live infants with grave physical or mental defects would have to be slaughtered. Family would be further undermined. The first generation of children so born would consider themselves as freaks. As for the monetary cost to create a human being in a laboratory and provide the necessary equipment, nutrients, and hourly monitoring by highly paid technicians would certainly run into thousands of dollars, and it is also unnecessary. The only social gain of any significance would be to free the working woman from having to interrupt her job in order to produce her baby. But for most working women, pregnancy is no problem at all for the first 7 months, and the womb gives a baby a vastly safer haven than any artificial womb could conceivably provide. I've known successful career women who stayed right on the job until labor started with no ill effects. But let's consider lifting the fetus from another mother at 6 months. If a working woman would prefer to have her baby removed, the incubator people are reaching the point where they can handle it. It would be expensive, but they could do it. The mother would be wiser to wait till the 7th or 8th month. The baby would be less at the mercy of man's ingenuity in manipulating knobs and nutrients. The earlier the infant goes into the incubator, the safe as it is becoming, the greater the statistical chance of death, defects, and mental impairment. The mother's womb will, for the great majority of people, always be the best, the healthiest place in which a baby can grow. Modifying our genetic blueprints. Chapter 16. This fair species of malleable man is to be created primarily by the strategies of geneticists and their scientific allies. It won't be created next year or in a decade, but a mere quarter century ago, very little was known about the mysteries of heredity. In that light, the recent breakthroughs toward manipulating our heredity equipment have been astounding. We literally have been thrust into a new biological epoch. The American genetic philosopher Theodosius Dobzhansky wrote in Science that emerging genetic technology would represent an instrument of scarcely unimaginable power for guidance of the evolution of the human species. But all is not celebration on the frontiers of genetics. At the International Congress of Genetics in 1973, the recitals of progress were interrupted by anxious questions and accusations of irresponsibility. Would a new eugenics lead to genetic discrimination against certain types of people? Would it provide subtle tools for social control? Soon thereafter, the scientific world was stunned when a group of the world's leading geneticists called for a moratorium on certain types of experiments in which genetic materials were being put into new combinations. Brand new creatures about which little was known were being assembled in laboratories. They were microscopically small, but some of them could possibly be deadly if they got loose in the world. There was long, restless pause throughout the world before such work was resumed under a cautionary code. But the dangers are still being argued. The geneticist James J. Nagel at Drew University points out that man is now on the threshold of consciously controlling and engineering his future in terms of the very composition of human population. That fact, he contends, raises dilemmas requiring decision. Some scientists are philosophically disturbed by any action that could put restraint on their innovative efforts. Robert Baugh-Miller, a geneticist at the Center for Bioethics of the Kennedy Institute in Washington, contends, if you're dedicated to truth, you have to say that there are no truths not worth seeking. Is creating a dangerous new life form a truth? For the immediate future, any effort to manipulate human genes by working on them directly will of necessity be on quite a small scale. The technical problems are fantastic, but there are strategies for modifying genetic endowment indirectly. These involve manipulating cells or modifying the cells containing the genes you have in mind, developing new kinds of human seed, selective breeding, eliminating or treating genetic defects at the prenatal, natal, and postnatal stages, and so on. We will get to them. But as a start, let's look at our incredible genes and the ingenious way that scientists are learning to modify them. How genes shape our bodies. No one had ever seen a gene until very recently. Even under enormous magnification, it shows up simply as a tiny, almost meaningless thread. Late in the 19th century, the Austrian monk and botanist Gregor Mendel worked out his universal laws on heredity for living matter. He deduced that there must be some physical substance that transmitted ancestral characteristics over generations, but he had no idea what it was. Early in this century, it was widely assumed that man's genetic endowment, however transmitted, was immutable. This was a comforting idea. It fell in 1927 when the geneticist Hermann J. Mueller reported that he had changed heredity patterns by the use of X-rays. Mutations showed up in this next generation. This evidence led him to advocate exploiting the apparent malleability of man by changing man for the better part through genetic manipulation. What the heredity transmitting material was remained a mystery. As microscopes became more powerful, scientists began assuming that thin strands called chromosomes, which they found in the nucleus of every cell, probably had something to do with heredity. Then in the mid-1940s, experiments by Aldwell T. Avery at the Rockefeller Institute produced a stronger clue. He concluded that the molecules of an obscure acid known as DNA, deoxyribonucleic acid, which was found in the strands of chromosomes, seemed to have something to do with heredity. Just how DNA was involved and how it performed its miraculous chores remained wholly unclear until the 1950s. Two flamboyant young biologists at Cambridge University, an Englishman, Francis Crick, and an American, James Watson, put many clues together and constructed a giant architectural model of DNA molecule. This was the master molecule of life, invisible except under strong magnification. But in their laboratory model, it reached from floor to ceiling and looked literally like a spiraling staircase. It was made of heavy wire, metal strips, and knobs. If they had attempted to construct an entire DNA molecule, it would have reached up through the ceiling and gone higher than a skyscraper. But their sample was enough to depict their well-informed guess. They called it a double helix. The two twisting banisters in a real DNA molecule, they explained, are made of phosphates and sugars. And each stair tread is made of two chemicals separating at the center of the tread and lightly held together by a hydrogen bond. Although, altogether, only 4 organic compound bases make up the treads of the spiraling staircase. They are adenine, guanine, thymine, and cytosine in various combinations, called for short AGTC. They constitute the genetic code. As Jean Rostand, a French biologist, eloquently put it, the properties of an individual's heredity depend on the way in which those 4 bases are arranged and ordered in their molecules. All the genetic diversity of the species stems from them, just as all our literature is written with 26 letters and all our music with 7 notes. Within a DNA molecule, a particular gene is the stretch of genetic code that is required to call up all the building blocks, amino acids, needed to produce a protein, often an enzyme, for a particular purpose in a bodily structure or function. These master DNA molecules contain the genes are found in every cell of a human embryo. And how does all this information get passed on as the future human body grows? Yes, the answer is one of the great wonders of nature. When a cell divides, this spiral staircase splits right down the middle of the treads along the line of the weak hydrogen bond. In a newly formed DNA molecule, each half of the old molecule picks up from the free-floating materials the ingredients it needs to construct its exact complement. Soon there are 2 spiral staircases where there was 1. And the dividing continues. These extraordinarily long, thin DNA molecules carry master plans for building a unique human being. And the plans get passed on to the next generation by way of germ cells. eggs, and sperms. A thimbleful of male semen contains tens of millions of sperm cells, and each sperm carries half of the male's DNA in its chromosomes. Likewise, each female's eggs carries half of her DNA. In a fertilized egg, the combined DNA provides the master plan for the new individual, complete with specifications for freckles, a weak chin, premature baldness, and a brain of a certain physical magnitude. Nature's achievement is comparable to printing the Bible on one's fingernail. The task of geneticists who aspire to delete or replace genes directly is at least as difficult as making editorial corrections on that fingernail. Crick and Watson won Nobel Prizes for their conception of the structure of the DNA molecule. Their concept was soon substantiated as essentially correct by laboratory experiments. It remained for another little-known American biologist, Marshall Nurenberg, of the National Institutes of Health, to crack the genetic code. In 1961, he matched the components of DNA with their functions and demonstrated how the building blocks get called up as needed. By the 1967, a strand of DNA was actually synthesized in a test tube. This was the feat of Arthur Kornberg of Stanford University. And ever since then, there have been fast-breaking achievements in getting to know and modify the gene. In 1970, the first complete gene was synthesized. This was the yeast gene, involving a stretch of DNA 77 base pairs long. It was put together at the Massachusetts Institute of Technology by a team headed by Indian-born biologist Har Gobind Khorana. By 1975, the first gene of a mammal, rabbit, was totally synthesized, this by a Harvard biologist. And in 1976, Har Gobind Khorana was back in the news again. His group had put together a gene out of shelf chemicals that actually did its work as a gene when implanted in a bacterial cell. This new gene had the crucial start and stop signals that in a living cell make a gene function. The gene was a linear sequence of 199 components, all involving a combination of the 4 ingredients of the genetic code, AGTC. The genetic mechanism was just as Crick and Watson had surmised 20 years earlier, mapping the genes. Meanwhile, the dozens of laboratories, geneticists began trying to pinpoint the location of individual human genes on man's 23 pairs of chromosomes. This knowledge is crucial if change or repair is to be attempted on a particular functioning gene. The main achievements in mapping have been performed by a team of 30 geneticists, biochemists, and cell biologists at the Yale University, headed by an amiable, lean giant named Frank Ruddle. In 1970, his group found their first human gene. Its function was to carry for the production of certain enzyme. By 1977, they had located more than 200. They had not only found which chromosome the genes were on, but in many instances, approximately where on the chromosome they were located. Ruddle and his colleagues do this mapping, not by studying specific genes, which is still impractical, but by studying the enzyme product created by each gene. They diagnose the area on the chromosome where the action that created the product was triggered. Ruddle hopes that by 1980, his group will have mapped at least 1,000 human genes. That still is only a small fraction of the 50,000 or so genes now believed to exist on human chromosomes. But some of those already mapped are known to be implicated in serious genetic disorders such as Tay-Sachs disease and the Lesch-Nyhan syndrome. The kind of analysis required to map the genes would not have been possible except for another major development. This permits genes to be manipulated by fusing 2 different cells. The fusion technique was developed after it was discovered that a virus called the Sendai virus will fuse cells. Cell fusion was pioneered by a team headed by George Barsky at the Institut Gustave Roussy in Paris. Mouse cells were fused. This was in 1960. By 1967, Mary Weiss and Howard Green at New York University were fusing the cells of mice and men. Fusing the cells of mice and men appealed to the Yale gene mapping team. When these newly created hybrid cells were placed in a nutritive broth, they divided in the usual way cells do. After many divisions, all of the mouse chromosomes were made, but only certain of the human chromosomes remained. This was apparently not due to any dominance of mouse over man, but rather to the differences in the rates at which cells of the two species mature and divide. The systematic isolation of different human chromosomes and the order in which they were shed made it easier for a mapping team to spot where actions of specific genes originate.